So our topic was topic 5, which was microbial control and epidemiology. We are group A7. So I was in charge of physical methods. There are various physical methods for microbial control that can be used to destroy or remove microbes. Some ways that include high pressure treatment, heat treatment, filtration, and irradiation. So high pressure treatments up to 120,000 psi may denature proteins and alter the permeability of the cell. Dry heat destroys cell components and denatures proteins. Incineration burns cell components to ashes. Dry heat oven less efficient than moist heat, requiring longer times in parenting. Moist heat denatures proteins, is relatively fast, reliable, safe, and inexpensive. Examples of moist heat treatments include boiling. So boiling for 5 minutes destroys most microorganisms and viruses. A notable exception is endospores. Pasteurization significantly decreases the number of heat-sensitive microorganisms, including spoilage, microbes, and pathogens, except endospore formers. Pressurized steam or autoclaving. Typical treatment is 15 psi above atmospheric pressure, 121 degrees Celsius, for 15 minutes or longer, a process that destroys endospores. And then I added an illustration here from the textbook, and it is an illustration of the autoclave. The steam first travels in an enclosed layer or jacket surrounding the chamber and then enters the chamber, displacing the air downward and out through a port in the bottom of the chamber. Another method is filtration. Filters retain microbes while letting the suspending fluid or air pass through small holes. Filtration, there are two types. Filtration of fluids, which is various pore sizes are available. The 0.2 is commonly used to remove bacteria, and the filtration of air. HEPA filters are used to remove microbes that have a diameter of 0.4 or greater. And then I added another image here, which is sterilization of fluids using a membrane filter. The liquid to be sterilized is pulled through the filter by means of a vacuum produced by a pump. The inset in the figure is a scanning electron micrograph showing a bacteria cell retained by a membrane filter. And then irradiation, which is a type of cell damage, depends on wavelength of the radiation. So ionizing radiation destroys DNA and possibly damages cytoplasmic membranes, produces reactive molecules that damage other cell components. Ultraviolet radiation, which damages DNA and penetrates poorly.
Chemical methods of control use any of a variety of antimicrobial chemicals that damage cells by denaturing proteins, altering DNA, or disrupting cell membranes. Choosing this method of microbial control is dependent on circumstance and level of control required. This is because some control measures adequate for daily life might not be sufficient for settings like a hospital, a microbiology lab, and food or water production facilities. Germicidal chemicals can be used to disinfect and sterilize. They are generally less reliable in heat, but useful for treating large surfaces and heat-sensitive items. Think of how we disinfect our surface areas in lab. Some points to be considered when choosing a germicide include toxicity, activity in the presence of organic matter, compatibility with the material being treated. For toxicity, germicides are at least somewhat toxic to humans and the environment, so there must be caution used when choosing one. An example of this in a real-life application is the same germicides used to disinfect hospital materials are not the same germicides you need to use to disinfect surfaces at home. The germicides used in hospitals are much stronger and can be more toxic to humans. Activity in the presence of organic matter. Bleach and many other germicides react with the organic matter, losing their effectiveness in doing so. Other chemicals, such as phenolics, tolerate the presence of some organic matter. Compatibility with the material being treated. Items such as electrical equipment often cannot withstand liquid chemical germicides, and so gaseous alternatives must be used. Residue. Many chemical germicides leave a toxic or corrosive residue. If such a germicide is used to treat an item, the residue must be removed by washing. Cost and availability. Some germicides are less expensive and more readily available than others. For example, hypochlorite can be purchased in the form of household bleach, which is cheap and easy to find. But alternatively, there are other germicides that need specific chambers to keep it in, or may be much more costly. Storage and stability. Some germicides are sold as concentrated stock solutions, decreasing the required storage space. Environmental risk. Germicides that retain their antimicrobial activity after use can interfere with wastewater treatment systems. The activity of those germicides must be neutralized before disposal. Categories of germicidal potency. Sterilants. These destroy all microbes, including endospores and viruses when used properly. They are also called sporicides. High-level disinfectants. These destroy all viruses and vegetative microorganisms, but they do not reliably kill endospores. Most are simply sterilants used for short periods, not long enough to ensure endospore destruction. Intermediate level disinfectants. These destroy all vegetative bacteria as well as fungi and most viruses. They do not kill endospores even with prolonged exposure. Low-level disinfectants. These destroy fungi, vegetative bacteria, except mycobacteria, and enveloped viruses. They do not kill endospores, nor do they always destroy non-enveloped viruses. Both intermediate and low-level disinfectants are called general-purpose disinfectants and are used in hospitals for disinfecting furniture, floors, and walls.
Okay, so microorganisms and bacteria play a huge role in the preservation of perishable products. They're actually the main reason why we see food spoilage or moldy bread or any of those things. The preservation of food is about preventing or slowing down the growth of bacteria, such as bacteria, fungi, and other microorganisms. There are many ways to try and preserve your perishables. Notice how it says slowing down the growth and not stopping growth, because just because you refrigerate or freeze something, it does not mean you are stopping the growth of organisms. You are simply just taking away their needs to slow down the growth of bacteria. So just because you take these precautions doesn't mean your food's not going to spoil. All food will and can spoil at some point or another. Psychotrophs are going to be the main type of bacteria that we see in food spoilage because they love 20 to 30 degrees Celsius. So even when you refrigerate things or freeze things, you can still see these in your food, and these can still live in your food and spoil your food because ultimately they can live in colder temperatures. Again, too cold, if you refrigerate it, it just simply slows it down, but does not kill them off or stop it. Just like us, organisms and microorganisms, they all need nutrients and things that they need to survive, one of them being water. So one way you can preserve your food is adding salt to your food, which can draw out the water, which decreases the bacterial lifetime because, again, they need water to survive. It also slows down the growth of other bacteria. So while it can decrease bacterial lifetime, that doesn't mean that they're going to die off and your food's not going to spoil. That's not true, because there's still other bacterias and they can still spoil your food, but it will slow down the growth of those other bacterias. Vacuum sealing is another way to prevent oxidation. It's usually used for meats and veggies. Oxygen's another thing that microorganisms and bacteria need to survive. So vacuum sealing your food will again prevent oxidation. So prevents prevents oxygen from getting into your food, therefore giving nutrients and oxygen to those bacteria, which can cause them to reproduce and spoil your food faster. So vacuum sealing is a great way to prevent oxidation. Again, refrigeration and freezing is good. I know a lot of people that freeze food because it ultimately lasts longer, and it's not a myth, it's true, it does last longer, and this is because the coldness will slow down the growth of bacteria. So again, does not eliminate the bacteria, does not stop it. You can still spoil your food this way, but it does slow it down. Placing foods in jars or similar containers and heating them to a temperature that destroys microorganisms that can cause food to spoil. So by heating up, a lot of bacteria can't survive certain heats and temperatures. Therefore, if you take a food and you throw it into a jar or container and you microwave your food, it can kill off a lot of the bacteria. And can destroy microorganisms that can cause your food to spoil. Again, your food can still spoil because new microorganisms can grow onto it, or if water gets inside your food, then microorganisms and bacteria love water, therefore they can get into your food, reproduce, ultimately spoiling your food. The fermentation process involves the oxidation of carbohydrates to generate a range of products. This helps with food preservation by limiting the growth of microorganisms. So the fermentation process helps preserve food by slowing and limiting the growth of microorganisms, but it does not stop it. So it uses oxidation of the carbohydrates to generate a range of products, and then ultimately just slows down the growth for those bacteria and microorganisms. So although your food will still spoil and can still rot and will still go bad at some point, there are many ways that you can save your foods or make them last longer, and hopefully that way we can stop wasting so much food and throwing away food. So if you know you're not going to eat something for a while, just throw it into the refrigerator or the freezer and just follow a lot of these precautions.
Antibiotics like B-lactams or penicillins hinder the formation of peptidoglycan in bacterial cell walls, causing cell lysis. Drugs like polymyxins disrupt bacterial cell membranes, leading to leakage and cell death. Quinolones interfere with DNA replication and repair, inducing bacterial cell death. Sulfonamides and trimethoprim disrupt folate production, which is crucial for nucleotide synthesis, impacting bacterial DNA and RNA. Antibiotics such as aminoglycosides, tetracyclines, and macrolides target bacterial ribosomes, hampering protein synthesis and causing bacterial dysfunction. Antibacterial medications like B-lactam antibiotics, glycopeptide antibiotics, and bacitracin interfere with peptidoglycan synthesis, weakening the cell wall and potentially causing the bacterial cell to burst. These drugs are most effective against actively growing bacteria since they continually produce peptidoglycan. B-lactam antibiotics, including penicillins, cephalosporins, carbapenems, and monobactams, all share a B-lactam ring. They are commonly used to treat bacterial infections because of their effectiveness and safety. These antibiotics work by disrupting the construction of bacterial cell walls, inhibiting enzymes known as penicillin-binding proteins. This interference weakens the bacteria, making it easier for the immune system to eliminate them. Some medicines stop bacteria from making proteins. The bacteria have something called a 70S ribosome, which is different from the 80S ribosome in our cells. The difference makes the bacterial ribosome a good target for these medicines, allowing them to harm bacteria without affecting our cells too much. However, because our cells' mitochondria have ribosomes similar to bacterial ribosomes, some of these drugs can still be a bit harmful to our cells. Some medicines, like sulfonamides and trimethoprim, interfere with the pathway in the body that leads to the production of folate. Folate is crucial for making a coenzyme needed to build DNA when you combine these medicines in a medication called cotrimoxazole. They work better together. Animals don't have the enzymes for this pathway, so they need to get folate from their diet.
So these are the most common antifungal medications. First off, you have polyenes. Polyenes bind to ergosterol, which is an essential part of fungal cell membranes, forming holes that damage membrane integrity. This results in intracellular substance leakage, which eventually kills the fungal cells. And then we have azoles. Azoles block linastrol 14 alpha demethylase, which is an enzyme that impacts the formation of ergosterol. And then we have allylamines. Allylamines inhibit squalene epoxidase, which is another enzyme involved in ergosterol biosynthesis, resulting in the buildup of poisonous squalene and damage to fungal cell membrane function. And then we have fluoxetine. This antifungal drug turns 5-fluorouracil within the fungal cell. It prevents RNA and DNA synthesis by blocking thiamidylate synthase, which disrupts nucleic acid metabolism and causes fungal cell death. And then we have griseofulvin. This antifungal agent disrupts the mitotic spindle formation by binding to microtubules, which inhibits fungal cell division and is commonly used to treat dermatophyte infections of the skin, hair, and nails. And then we have echinocandins. Echinocandins inhibit the enzyme beta-1,3-D glucan synthase, which plays a role in the production of beta-glucans in fungal cell walls, which weakens it. And then we have benzimidazole. These drugs interfere with the function of fungal mitochondria, leading to the disruption of energy production and cell death. So why are antifungal medications important? So these are critical in treating a wide range of fungal infections, from minor skin concerns to life-threatening systemic diseases. These medications are critical due to the increasing number and possible severity of fungal infections, especially among immunocompromised people. Fungal pathogens can cause pain, deformity, and even life-threatening problems. As a result, antifungal treatment is critical for improving patient outcomes. The rising frequency of diseases such as HIV, AIDS, along with the widespread consumption of immunosuppressive medicines and long-term antibiotic treatments, have demonstrated the importance of these medications. Antifungal medications are an important line of defense, allowing healthcare practitioners to adapt treatments to the accurate type and severity of an infection. These drugs not only improve effectiveness, but also help reduce the chance of resistance, ultimately playing a vital role.
Guys, I'm Cameron Iverson, and I will be talking about Christian worldview of microbial control. So my first example that I found was ceremonial uncleanliness. The Bible states that when a woman gave birth, after this birth, she would go through menstruation for about 7 days, and she was considered unclean. We see this through Leviticus 12:2. A woman who becomes pregnant and gives birth to a son will be unclean for 7 days. This means that she was segregated off from other people, as well as she was required to bathe more regularly. There was also a connection between this verse and Numbers 19:22. Furthermore, anything that the unclean person touches shall be unclean, and the person who touches it shall be unclean until evening. In the mid-1800s, Ignaz Semmelweis discovered the hand-washing technique in Austria. From this, Ignaz was observing doctors when he had noticed that they would go straight from handling diseased bodies in the morgue to delivering babies. After Ignaz introduced the hand-washing technique, the infant mortality or the maternal mortality rate went from about 15 to less than 5%. In this sense, the law of Moses was ahead of its time in protecting new mothers and babies, whilst also spiking the nation's ability to populate more rapidly. My second example that I have is the sanitation and management of human waste. So the verse that I chose for this was designates a place outside of the camp where you can go to relieve yourself. Deuteronomy 23:12. The toilets are a relatively modern invention, and they were not always around to help with the sanitary needs that we do need and the disposal of human waste. So due to the Israelites having a nomadic existence, they did not have the equipment to properly do underground water sewage. And with this, the Israelites had this rule of going to the restroom in a hole that they had dug for themselves outside of their camp. And after they would use the restroom, they would cover up this hole afterwards. And it has been proven that if this technique were further practiced before modern times of the toilet, this technique would have prevented the spread of many gastrointestinal diseases that were spread through unsanitary restroom oases. My third and final example of Christian worldview of microbial control is food hygiene through the laws of Moses. So I found this verse of Leviticus 19, 5-7. When you sacrifice a fellowship or offering to the Lord, sacrifice it in such a way that it will be accepted on your behalf. 6, it shall be eaten on the day you sacrifice it or on the next day. Anything left over until the third day must be burned up. 7, if any of it is eaten on the third day, it is impure and it will not be accepted. Leviticus 19, 5-7. The laws of Moses also abide by rules that would have improved food hygiene by separating animals into a clean and an unclean category. The animals that were considered clean were allowed to be eaten. They consisted mostly of cattle, some birds, and fish with fins. The other category would be animals that were considered unclean and therefore were not allowed to be eaten. And these animals consisted of certain meats such as pork, carrion feeding birds, filter feeding fish, and most seafood. From a hygiene standpoint, it can be depicted that the foods allowed to be eaten were inherently safer than the foods that were considered unclean and should not be eaten. Another important note is that there is no refrigeration in the desert, especially back in these times. So further, the laws of Moses would have provided an expiration date for what could be fermented foods. Next, these are our sources. Thanks for watching.